This is the second lesson of Chapter 9. We're continuing to look at cell reproduction and growth. And in our first lesson, we learned about why cells are small and what limits the size of cells, what prohibits them from growing indefinitely. Ultimately, cells divide. As a cell continues to grow until it reaches its maximum size, it will then divide. So in this lesson, we're going to be looking at why, first of all, why this is a necessary process, cell division, and then we're going to go through the process together of cell division. So there's several reasons why cells divide. It could be for growth and development, just because an organism, as it's growing and developing, needs to make more cells to become larger, more complex. Now, this would only pertain to a multicellular organism, which would also have to be a eukaryote. So this does not pertain to a prokaryotic cell for growth. However, prokaryotes do use this mechanism for reproduction to make basically offspring or copies of themselves. And in addition, for eukaryotes, they will use it for repair to, replace cells that have been damaged or become missing, perhaps like because of a wound, like when you get a cut on your skin and those cells that were cut were either damaged or maybe even completely removed from the body. So the neighboring cells will begin the process of basically copying themselves to make identical cells to themselves and repair that wound. So there's many applications for cell division. Let's slide that up a little bit. Okay. So let's take a look at the step-by-step process that's used by a cell to make a copy of themselves. This is the mitotic cell cycle illustrated here. And I emphasize mitotic cell cycle because in Chapter 10, we'll learn about the meiotic cell cycle. There are some similarities to both of these, but there are also differences, including the end result. So for now, we're just going to focus on the cell cycle and related to mitosis. So notice that this cell cycle is, if you look at the center, there's interphase is listed here in the middle. And this interphase takes up about 90% of the cell cycle. After interphase is the mitotic phase, or mitosis, which goes hand in hand with cytokinesis. So the way that that's shown in different colors there, that wedge is meant to show that the cell cycle overall has these two major phases of interphase and the mitotic phase. Within each of these overall phases of interphase and mitotic phase, there are also sub-stages of each of those larger phases. Notice that interphase can be further broken down into the three phases, which are G1, which is the abbreviation for gap 1. So the first stage of interphase is gap 1 or G1, followed by synthesis or S, and finally gap 2 or G2. The mitotic phase includes 5 subphases, but one is simultaneous to a process of cytokinesis. So those are listed there as prophase, prometaphase, metaphase, anaphase, telophase, which is simultaneous to cytokinesis. Now we're going to go into each of these stages and look at them more closely, including making some drawings of a hypothetical cell going through these stages. But before we do that, I just want to mention what happens in the substages of interphase, because when we're looking at a cell in interphase, you can't differentiate which subphase it's in. So it is important to know, however, how the cell is progressing through interphase. In gap 1 or G1, all of the cellular contents except for chromosomes are duplicated. So any organelles that are present that are going to be a part of that cell and the daughter cell that results from the duplication. All of those other cell parts are going to be duplicated. Then in S phase, the chromosomes are duplicated. In the slide here, it says each of the 46 chromosomes is duplicated. Well, that's talking specifically about a human body cell. So that number 46 might differ depending on which organism this is happening in and what is their normal chromosome number. Okay, so just be sure you know that in the synthesis phase, this is when the genetic material or chromosomes are duplicated or synthesized, if that helps you remember. Finally, in GAP2 or G2, the final checkpoints are made, so any errors that might be caught can be corrected before those errors make it into the daughter cell. Okay, now we're going to go into each of the phases and highlight some of the key events and structures that would be present in those phases. You have a page in your course packet that has a way you can organize this information by doing a drawing and then making notes about the key events. So I'll be making the drawing here in this white circle on the right and narrating that when it's finished. Okay, so I've added some details to the drawing of a cell that's in interphase. This is a cartoony version, of course. In the top left corner of the slide, you can see an actual photograph taken with a compound light microscope of an animal cell during interphase. So now the animal cell that we're going to be drawing throughout the process is going to be one that has a normal chromosome number of 4, or you could say it has a diploid number of 4 chromosomes, or 2n equals 4, as notated below that cell I drew there. So the most important things to note not only are, what could be happening during interphase if it's in G1S or G2, as we just discussed, but what you can actually see if you were to observe one of these cells in interphase under a microscope. You would see the nucleus, and possibly you can see the dark spot inside, the nucleolus. And in the nucleus is, of course, the genetic material. In this form, it is chromatin, uncoiled, uncondensed DNA. It's not yet chromosomes until they condense in actually the next stage. So be familiar with the terminology. Here we have the nucleus with chromatin, uncondensed chromosomes. Now I also drew at the top a pair of centrioles in a centrosome. This structure is going to be responsible for forming the mitotic spindle that will help to coordinate the movement of the chromosomes in the later stages. Right now in interphase at this point, I would say it's probably in G1 because by the end of interphase, there would actually be a copy of that structure, the centrosome with a pair of centrioles. But again, you're not going to necessarily see that. As you can tell from the microscopic photograph at the top left, the only real notable structure is the nucleus and has a really distinct outer nuclear membrane. Inside it looks kind of just grainy, no distinct chromosomes yet. Okay, so let's see what happens next in our first stage of mitosis, prophase. So here we have a cell in prophase, and at the top left, you can see a different type of cell, a plant cell, several plant cells actually, in different stages of prophase. There is early and late prophase. It's really a continuous process, so depending on at which instant in time that organism or those cells are fixed, in the lab, or basically the cells are killed at a particular instant in time, is what you're going to see frozen in that slide or that illustration. So I'm going to be drawing, or I drew a cell that would be more in early prophase, like the cell shown in early prophase at the top left. So some things to note with that. First of all, let's talk about the spindle fibers and the centrioles in the centrosomes. So notice that the centrosome containing a pair of centrioles has duplicated, and they've started to actually diverge. And the green lines represent the spindle fibers that are forming between the two pairs of two centrosomes. And that will continue to lengthen. Those green lines, which are, again, they're spindle fibers and they're made of microtubules, a type of cytoskeleton of the cell. Now, where the nucleus was, the nuclear membrane has started to disintegrate, and the chromatin inside the nucleus has condensed into chromosomes. These chromosomes have been duplicated because, remember, by the end of gap 2, the genetic material has duplicated. So what we have here are actually duplicated chromosomes, or you could call them sister chromatids. That's important. We're going to call them sister chromatids while they're connected, basically like conjoined twins. You could think of it like that. But once they separate, we'll call them something else in a later stage. The next stage of mitosis is called prometaphase, which means before metaphase. So prophase involved the formation of the spindle, and now you can see the spindle has migrated basically across the whole length of the cell. The spindle fibers have connected to the pairs of sister chromatids, and they attach by a structure located at the center, the centrosome. There's special proteins there called kinetochore proteins that each of these sets are attached to the spindle fibers. Now, the ones that they're attached to have a specific name. These are called kinetochore spindle fibers or kinetochore microtubules. So that refers to any of the ones that have the pairs of sister chromatids attached to them. Okay, it's getting a little busy if I draw those lines. Let me take those off actually. Versus the non-kinetochore microtubules are the microtubules of the spindle that are not connected to any genetic material. They're just basically spanning the entire length of the cell. So I'll highlight those in a different color, I think, and that will make it a little more clear. So to emphasize that, I just highlighted the non-kinetochore microtubules on the drawing and related that to the note here on the bullet points. Next is metaphase, which refers to the instant in time when the pairs of sister chromatids have lined up along the middle of a cell. There's like an imaginary line, like the equator of the Earth, called the metaphase plate. I'm going to draw that as a black dashed line. Just again, it's an imaginary line. It's not a real place, but it's where those pairs of sister chromatids line up. And then we have anaphase, which is when the pairs of sister chromatids become separated as their centromeres divide, and now we refer to them as daughter chromosomes. The daughter chromosomes are going to be pulled to opposite poles of the cell by the kinetochore microtubules. So the kinetochore microtubules are actually getting shorter and shorter as they migrate to opposite poles where the spindle fibers originate, the centrosomes, while the non-kinetochore microtubules of the spindle actually are lengthening. Notice the cell has been stretched a little bit. It's more like an egg shape than a round circle because of the non-kinetochore microtubules that are stretching out the cell. The final stage of mitosis is telophase. Notice the spindle fibers are no longer present. They've been reabsorbed by the cell, but each side of the cell that's now still attached but stretched out more and starting to kind of show the separation. Each of those has a pair of centrioles and a centrosome, as well as the 4 chromosomes that are now contained in a region where the nucleus is starting to reform, as shown by the dashed purple lines, representing the nuclear envelope beginning to reform again around that genetic material. So chromosomes are becoming less condensed. Again, it's a transitional phase, but we'll see that come back to interphase eventually. So let's take a look at what comes next. At the same time as telophase is cytokinesis, which is not technically mitosis. It is the process of the division of the cytoplasm. So all the while telophase is happening, cytokinesis is happening. The cells are physically dividing, becoming 2 independent daughter cells. So what I showed here would be more of like a late telophase because the nuclei have reformed. You can see a nice nuclear envelope, and inside is no longer chromosomes, but it's the uncondensed chromosomes, which is now referred to as chromatin.